Welcome to The Explainer. Today, we're diving straight into the absolute fundamental architecture of life. We're talking about prokaryotes versus eukaryotes. Honestly, whether you're looking at a towering redwood tree, a microscopic germ, or just catching your own reflection in the mirror, it all ultimately comes down to this one major structural fork in the road of biology. So I want you to start by asking yourself, what actually makes you different from a bacterium? I mean, sure, there's the obvious stuff, right? You have a brain, you can walk, you're listening to this explainer right now. But if we zoom all the way down to the microscopic level, how does your incredibly complex cellular makeup fundamentally differ from those tiny microscopic bacteria that literally still rule our planet today? Well, to figure that out, we really have to look closely at the blueprints. Which brings us to Act 1, the rules of life. Before we can start comparing these cells, we absolutely have to establish the ground rules for anything to be considered alive in the first place. You can kind of think of this as the constitution of biology. And here they are, the three foundational principles of cell theory, originally laid out by scientists Schleiden, Schwann, and Virchow. Number 1, all organisms are composed of cells. Period. Number 2, the cell is the basic unit of structure and function, meaning it's the absolute smallest unit that can actually conduct metabolic activities. And number 3, cells come only from pre-existing cells. Basically, they have to divide to pass on their hereditary information, their DNA, to new daughter cells. Okay, so these are the absolute laws of biology that are going to ground our entire investigation today. All right, let's move right along to Act 2, the lineup, because every single living thing on Earth is going to fall into one of two main cellular categories: prokaryotic and eukaryotic. And here they are, our two suspects side by side. Over on the left, we've got the simpler, kind of pill-shaped prokaryotic cell. This represents organisms like your everyday bacteria. And then right next to it on the right is the highly complex eukaryotic cell, specifically an animal cell in this case. Just taking a quick glance here, you can already see a massive, stark contrast in just how busy the insides of these cells actually look. Let's get into Act 3, spot the difference. It is officially time to turn you into a cellular detective. I'm going to guide you through a little visual investigation here, so definitely get ready to actively scan and spot the critical differences that separate these two architectures. Okay, here's your very first assignment. Every single cell needs a blueprint, right? Its genetic material to survive and function. Try to figure out exactly where that genetic material is being stored in both of these cell types. Take a really close look here. Over on the right, we have a eukaryotic plant cell, and on the left, our prokaryotic bacterium. Do you see where the DNA is hiding? I'll give you a second to just kind of scan the diagrams. You're probably noticing a pretty massive difference in how the center of each cell is actually put together. So here is the absolutely crucial point. Prokaryotes literally just leave their genetic material completely unenclosed. It's just hanging out, lying freely in the cytoplasm, in a region that we call the nucleoid. But eukaryotes, they protect their DNA inside this incredible high-security vault called a membrane-bounded nucleus. This nuclear membrane effectively walls off the genetic code from the rest of the cell. It's a huge difference. All right, so we've found the vault. Now let's pose our next investigative question. How is the actual DNA blueprint physically organized inside each of those spaces? Because, having a secure vault totally changes how you're going to store your valuables. Now, check out the precise contrast in the DNA itself. Prokaryotes, on the left, keep things simple. They've just got a single circular piece of DNA, and it just floats around without needing any special structural proteins. But eukaryotes, man, they have to manage massive, highly complex amounts of linear DNA. So to keep it all from just tangling up into a huge mess inside that nucleus, the linear DNA associates with histone proteins. It actually wraps around them almost like thread around little spools. It is just a beautifully organized system. Okay, we have one more major clue to track down, but before we hunt for it, let's explicitly define a super important term, an organelle. You can really just think of organelles as specialized factory departments inside the cell. Each one is enclosed by its own membrane, and each one is doing a very specific, crucial job to keep the whole cell alive. So, keeping that exact definition in mind, I want you to scan the insides of both cells again and ask yourself, did you catch what's completely missing from one of these setups? Let's pause here for a second so you can really look. Scan that eukaryotic cell on the right. You're going to see membrane-bound structures practically everywhere. I mean, there's the mitochondria pumping out power, the Golgi apparatus acting like a packaging center. Now, look over to the left at the prokaryotic cell. Do you see any of those specialized membrane-enclosed factory departments in there? And our investigation officially confirms it. Prokaryotic cells entirely lack membrane-bounded organelles. Their internal space is just totally open. Eukaryotic cells, however, are incredibly compartmentalized. They've got tons of membrane-bounded organelles that perfectly divide up the cellular labor. Which brings us to Act 4, the big picture. Okay, so we've successfully spotted the three major differences, right? The nucleus, the DNA structure, and the presence of organelles. But let's zoom out for a second. Why does this cellular architecture actually matter in the grand scheme of biology? It matters because architecture completely dictates capability. The advanced setup of eukaryotes, having that protective nucleus, the superorganized linear DNA wrapped around histones and all those specialized organelles, it allows them to be vastly larger. I mean, while prokaryotes sit at a tiny 1 to 10 micrometers, eukaryotes can easily be 10 to 100 micrometers. And that internal organization, that is exactly what allows eukaryotic cells to join together and form incredibly complex multicellular life. We're talking protists, fungi, plants, and yes, animals like us. Which brings us right back to you. Now, those simple, uncompartmentalized bacteria, they are undeniably successful. They've been here since the literal dawn of life, and honestly, they still rule the Earth in sheer numbers. But without that membrane-bounded nucleus, without those histones organizing the DNA, and without those specialized organelles, life just could never have scaled up. So I'm going to leave you with this to ponder. How did the sheer architectural leap from those simple, floating nucleoids eventually build the trillions of highly organized eukaryotic cells that precisely coordinate to make up the complex human being you are today?